Etem?
Yeah?
I was just wondering, do we still need to do our left-hand page for bio?
What?
The thing for bio. The photosynthesis and cellular respiration page thing?
Yeah, what about it?
I was just wondering if we still had to turn it in and present it. Well, hello, and welcome to my presentation on photosynthesis and cellular respiration. So, let's first talk about photosynthesis. We all know that in order for photosynthesis to work, we need sunlight, water, carbon dioxide, and all that good stuff, right? Well, first needs the light to enter the leaves of the plant, then through the thylakoids of the chloroplasts and into a protein complex called photosystem II. All of those electrons within photosystem II then become excited and leave traveling down the electron transport chain.
Wait. What do you mean by excited?
Oh, that means they're gaining energy.
Alright, go on.
After that, a water molecule is split. This is to replace electrons lost from photosystem II. This also creates hydrogen ions and oxygen. Now, the electrons that were traveling the transport chain from before are used to pump these electron ions through the thylakoid membrane and into the thylakoid space.
What's another name for ions?
Protons. And due to the increase of H+, there is a higher concentration in the thylakoid space and a lower concentration in the stroma. Diffusion allows the H+ to move into the areas of lower concentration, and the ATP synthase protein allows this to happen. The H+ causes it to rotate producing ATP. This ATP will then be used as an energy source to create sugar during the Calvin cycle. Now, do you remember the electrons from before? The ones in transport chain? Well, now they're in photosystem I. Light strikes photosystem I similar to that of photosystem II, and the electrons become excited once more. Then they go down another transport chain in order to reduce NADP+ to NADPH, which carries these electrons into the stroma for the Calvin cycle.
Okay, you've mentioned the Calvin cycle twice now, and you've explained what is given to the Calvin cycle. Could you explain what it does?
Oh yeah! In the Calvin cycle, there are 3 phases. In the first phase, 3 RUBP molecules are combined with 3 CO2 molecules by the enzyme Rubisco in order to make 3-triphosphoglycerate.
How many triphosphoglycerates are made after adding CO2?
2 are made from every RUBP and CO2 pair. And the second phase reduces the triphosphoglycerate using the ATP and the electrons from NADPH from before to make 6G3P. 1 G3P then exits the Calvin cycle and is used in order to make organic compounds like glucose. Then in the third phase, the other 5 G3P is used to recreate RUBP so the cycle can continue. In order for this to work, it requires some ATP from the light reaction before. The Calvin cycle then needs to repeat phase 1 and 2 in order to release another G3P to combine with the G3P from before in order to create glucose.
Aren't you forgetting something? You only explained photosynthesis.
Oh yeah, cellular respiration. I almost totally forgot about that.
Could you make it quick and informational? It's getting dark out.
Okay, but why? Oh my god, are they trying to get in?
It's okay. Continue, I'm listening.
Okay. Cellular respiration has 4 stages: glycolysis, the link reaction, the Krebs cycle, and the electron transport chain. Glycolysis happens with the cytoplasm of the cell. Here, ATP is used to split glucose into 3 carbon molecules and produce 2 ADP molecules. 2 NAD+ takes the electrons from the 3 carbon molecules and becomes 2 NADPH, oxidizing the molecules. The phosphates from the 3 carbon molecules are then transferred to the 4 ADPs, which then turn into 2 pyruvate molecules and 4 ATP molecules.
Speed up a bit.
During the link reaction, pyruvate is oxidized, having its electrons taken in order to create 2 NADH, and is turned into 2 CO2 and 2 acetyl CoA. The Krebs cycle is within the mitochondrial matrix. Within the Krebs cycle, the acetyl CoA reacts with the acetylacetate, creating a citric acid. The citric acid then oxidizes by NAD+ to make a 5-carbon molecule, then again with another NAD+ to make a 4-carbon molecule, 2 CO2 and 2 NADH. This 4C molecule is then used to combine ADP with phosphate creating ATP. Then the 4C molecules oxidize by FAD, making FADH2, which reacts with NADH and another NAD plus in order to recreate axial acetate.
One more stage and then we can get out of here.
The last stage for cellular respiration is the electron transfer chain. This occurs within the inner membrane of the mitochondria. A NADH molecule transfers its electrons to the first protein of the electron transfer chain, and that energy from the electrons is used to pump the hydrogen ions into the inner membrane space. FADH2 then transfers some of its electrons into the chain, and then accumulated electrons move along the transfer chain, pumping in even more hydrogen ions 2 more times. This creates a high concentration within the inner membrane space. The electrons then attach themselves to the oxygen and the hydrogen that were in the matrix making water. The hydrogen ions then go through diffusion through ATP synthase, producing ATP. It also produces 30 to 34 ATP per glucose entered.
Run!
Go. So, how was the presentation?
Yeah, it was good. Yeah, I'd say it was a 10 out of 10.
A 10 out of 10? Cool. Since when were they trying to get in?
I'd say right after the first half of photosynthesis, one almost actually got you.
Wait, what?